Today I will explain some basic concepts regarding coelom. Coelom is a cavity or coelom is a body cavity which is lined by mesoderm on all the sides. We can say coelom is derived from mesoderm and filled with coelomic fluid. So coelom is a fluid filled body cavity which is completely lined by mesoderm and it is present between gut and body wall. On the basis of body plan, organisms can be classified as diploblastic organisms or triploblastic organisms. As the name suggests, in the diploblastic organisms, there are developmentally two germ layers. The first one is ectoderm, which will form the body wall, and second is endoderm, which will form the gut wall. Whereas in case of triploblastic organism, there is additional layer present in between the ectoderm as well as endoderm, and we call that layer mesoderm. And this mesoderm will lead to formation of body cavity, and such a body cavity which is surrounded by all the sides by this mesoderm and filled with coelomic fluid, we call such body cavities as coelom. And on the basis of whether the coelom is present or not, the triploblastic organisms can be further classified into acoelomates if coelom is absent and coelomates if coelom is present. And if we talk about what is the role of this mesoderm, why do we need the mesoderm in the first place? So there are two roles of this mesoderm. First one is to form the body cavities, and we will talk in great detail what is the significance of these body cavities in upcoming videos. And second role it serves is formation of peritoneum, which will line the internal organs which are going to form as per evolution. So this is the basic concept of the coelom. Let's start with the acoelomate organisms, in which a fluid filled body cavity lined by mesoderm is completely absent. There is no body cavity, as you can see. So this is a triploblastic organism, and we are taking the example of flatworms. Outermost layer, as you can see, is ectoderm, which is forming the body wall. Innermost layer you can see is endoderm, which is forming the gut wall, and in between the ectoderm as well as endoderm there is a layer we call mesoderm, and mesoderm is completely packed, completely filled. There is no body cavity which has been formed. As you can see in this body plan, the outermost body wall which is ectoderm, there is innermost gut wall which is endoderm, and there is the filling of mesenchyme parenchymatous tissue. There is solid filling. So we call these organisms having a solid body internally. What is the role of this mesenchyme tissue? So mesenchyme tissue is a tissue of loosely packed cells, and it functions in the support, transport and regeneration, as we will see in the planarian regeneration. Which organisms can be considered as acoelomates? The most common examples are first triploblastic organisms, which is platyhelminthes, which are dorsoventrally flattened, or we can say flatworms. All the flatworms are typically acoelomates. Examples you can remember as planaria. There is Taenia solium, fasciola is what we call liver fluke, and Taenia solium is what we call the tapeworm. In the upper diagram, we can see the acoelomate body plan. The outermost layer, colored layer, is the body wall, we call epidermis, or we call specifically ectoderm. The innermost layer, which is lining the cavity of gut, is gut wall, or we can say endoderm. And between this endoderm and ectoderm, there is solid filling, we can call this mesoderm, and there is no body cavity visible. We will talk about this lower figure later. What are the characteristic features of acoelomates? There is no body cavity. If there is no body cavity, there is no space for fluid based circulation, and there is no specific organ suspension. As you can see, gonads are lying freely in this solid filling of mesoderm. So there is no body cavity, the fluid based circulation is entirely absent, there is no specific organ suspension, organs are freely suspended in the mesoderm. And advantages of acoelomate body plan is that it is providing structural stability because the body is solid internally. There is efficient diffusion over short distances. So these organisms, all the flatworms, are typically smaller in structure. There is the higher ability of regeneration in some forms like planaria. But are the limitations of this acoelomate body plan? The first limitation is obviously the size constraint. So diffusion is only for the short distances, so there cannot be a very large body. They are very small organisms. There is no hydrostatic skeleton, so movements are less effective as compared to other proper coelomate organisms. There is limited organ complexity, so organs have to be freely suspended. As you can see, the gonads are freely suspended in this mesenchyme tissue. Finally, there is transport insufficiency, because first the diffusion is over very short distances, and there is no specific circulatory mechanism, so there is no internal fluid circulation, so transport will be very much inefficient. So there is size constraint, the organism size has to be very small, there is no specific hydrostatic skeleton, there is limited organ complexity, and there is no mechanism of specific transportation. So acoelomates are primitive kind of organisms, and examples we can take all the examples of phylum Platyhelminthes. If we talk about other organ systems, a digestive system is open type digestive system. There is only one opening for ingestion and egestion of food. If we talk about a respiratory system, respiration is through diffusion, and diffusion is across the body surfaces. There is a ladder-like nervous system if present at all. Now let's talk about primary coelom. If the body has to evolve some kind of body cavities, it was the primary coelom which was evolved first, sometimes also referred as pseudocoelom. Primary coelom is a body cavity which is derived from blastocele. That is the major difference. Coelom was the body cavity which was derived from the mesoderm itself. So in case of primary coelom or pseudocoelom, it is derived from the blastocele. It is not lined by mesoderm from all the sides, and typically outer side is lined by mesoderm and inner side is lined by the endoderm. And examples we can take nematodes, roundworms. Example is ascaris. We can take some rotiferans as well. What is the difference between the terms primary coelom as well as pseudocoelom? Primary coelom, if we talk about primary coelom, we are talking about the developmental process. So it has developed from the blastocele. So it is blastocele derived cavity. And if we talk pseudocoelom, we are talking about structure specifically, that it is not completely lined by mesoderm. If we talk about structure, we can say it is pseudocoelom. And if we talk from where it is developed, we can talk in the terms of primary as well as secondary coelom, because the primary coelom is what is derived from the blastocele, and secondary coelom is what is derived from the mesoderm itself. Outermost layer is obviously ectoderm, and innermost layer is endoderm, which is gut wall, and outermost is body wall. In between these layers there is mesoderm, which is shown in green. Mesoderm and gut wall has another cavity, pseudocoelom, because it is outwardly lined by this mesoderm and inwardly lined by endoderm, as you can see. So it is not completely lined by mesoderm, so we cannot call it as complete coelom. Pseudocoelom can act as some kind of hydrostatic skeleton, but it will not function that efficiently which proper coelomic hydroskeleton works. It helps in the distribution of nutrients, transportation of waste products, because the circulatory system has not evolved yet. So what are the limitations of pseudocoelomates? So first one is they lack true organ suspension. So organs again lie in the free pseudocoelomic fluid. There is no specific formation of mesenteries on which these organs can lie. They will lead to poor spatial organization. The second point is they have limited control over organ movement because they are lying in this free suspension, and there is no independent movement of gut and body wall. Both gut as well as body wall will have to be moved together. There is a reduced structural stability because the cavity is not compartmentalized. There is no mesenteries, no internal connections, and internal pressure is controlled in a less precise way. So hydrostatic function is considered somewhat primitive. There is absence of peritoneum, so there is no specific internal epithelial lining for internal organs. There is reduced secretion, absorption and protection as a result of this lacking of specific epithelial linings. The organs will be less specified, less specialized, and there will not be the formation of specific or complex structures. There is limited circulatory efficiency because it lacks the proper circulatory system. There will be diffusion. Diffusion will be more as compared to acoelomates, but still be less and passive as compared to true coelomates. And again, there is no compartmentalization. Organ isolation cannot be there. There will be limited physiological functions. There will be evolutionary restraints. So it represents a kind of intermediate or transitionary condition, and it does not support the large body size, which was the same limitation as we saw in case of flatworms, in case of Platyhelminthes, the acoelomates. Now let's talk about secondary coelom. If I'm saying secondary coelom, I'm talking about the developmental perspective, and if I talk about structural perspective, I will have to say it as only coelom or eucoelom. or eucoelomate body plan. So secondary coelom is a body cavity which is found within mesoderm. It is found within mesoderm, so it is lined by mesoderm from all the sides. Blue colored, it is ectoderm body wall. Innermost, which is slightly pink colored, which is endoderm, and in between endoderm and ectoderm there is this green colored layer which is mesoderm, and there is a cavity which is lined by mesoderm from all the sides. This is what we call as coelom, and this coelom is filled with coelomic fluid, and there are mesenteries forming, which you can see right here, and these mesenteries will keep all the organs in their specific places so that the specific organizations and specific organs can be evolved. Examples: Annelida are the first true coelomates. The most common example, earthworm, arthropoda, in which insects, crustaceans, all those organisms will lie. Mollusca are also true coelomates. We have chordates, we humans, vertebrates. We also have this eucoelomate or true coelomic body plan as well. As you can see, outermost layer is epidermis, and we call this layer ectoderm. The gut wall is gastroderm, or we call this layer endoderm. Between this ectoderm and endoderm we have mesoderm. There are cross connections, cross bridges, and these cross bridges are what we call mesenteries. So we have dorsal mesentery on the dorsal side. This side is dorsal and this side is ventral, and we have ventral mesentery on the ventral side of this body, and this is the coelomic cavity which is filled by coelomic fluid, as you can see. And due to these cross connections, cross bridges, mesenteries, there is specific arrangement of organs, and organs can be specialized. As you can see, the gonads are properly supported in a retroperitoneal position. So this coelomic cavity is filled by coelomic fluid, and coelomic cavity is externally lined by the parietal peritoneum, which is derived from mesoderm, and it will line the body wall, and internally towards the gut it is lined by the visceral peritoneum, which will cover the various kinds of organs that are going to be formed along with this body cavity. So this is the basic structure of the coelomate body plan. Let's talk about mesentery. So mesentery is a double layer of peritoneum which is mesoderm derived. It functions as a suspension for gut and all the developed organs. It keeps all the organs on their specific place. That is the main function of the mesentery. Mesentery is what connects all these organs to the body wall itself. It is a bridge formed by folding of mesodermal lining, and mesentery is formed by the splanchnic mesoderm. If we talk about the actual developmental fate, functions of this coelomic cavity, so it is providing a proper hydrostatic skeleton. It is providing independent organ movement. There has to be no synchronous movement between the gut wall as well as body wall. There can be independent movements, which was the main limitation in case of lower organism like acoelomate, flatworms. There is efficient organ development, there is a specific organ development, there is specific compartmentalization because the mesenteries have already been developed. There is a proper circulatory system. So what are the differences between primary and secondary coelom? If we talk about origin, primary coelom was originated from the blastocele itself, whereas secondary coelom was found within the mesoderm. And if we talk about mesodermal lining, it is incomplete. It may be externally lined by mesoderm but internally lined by endoderm, whereas in case of complete coelom or specific secondary coelom, it is lined by mesoderm from all the sides. If we talk about peritoneum, peritoneum is not developed in case of primary coelom, and same goes for the mesentery. So peritoneum and mesentery are only specific for the secondary coelom, and the organ suspension will be poor. There will be primitive type of organs in case of primary coelom, and organs can be well developed, well specialized in case of secondary coelom. And if we talk about evolutionary perspective, secondary coelom is more recent, more advanced. Now let's talk about this fluid which is filled inside the coelom, or even in case of pseudocoelom, the fluid which is filled is what we call pseudocoelomic fluid. So fluid presence is in both cases pseudocoelomates as well as eucoelomates, and control is more regulated in case of eucoelomates. There is specific peritoneum lining, and mesenteries are also present in case of eucoelomates. Coelomic fluid is originating from the process of ultrafiltration of blood or interstitial fluid, and after this ultrafiltration process, it is modified by the mesodermal coelomic epithelium, which is originated from the peritoneum itself. Now let's talk about the developmental origin. Coelom is formed by two processes. If the organism is protostomous, it is formed by the schizocele process, and if it is deuterostomus, deuterostomus. It is formed by the enterocoelom process. I will make a separate video on the protostomes as well as deuterostomes. And for now, the protostomes are the organisms in which the mouth is formed first, whereas in case of deuterostomes, the anus is what will be forming first in the developmental process. And initially, the cavity is filled by extracellular fluid, and this fluid is later converted into coelomic fluid. And if we talk about physiological origin, it is only present in case of adults. And there are three main processes which are responsible for the physiological formation of this coelomic fluid. First one is again the same process, ultrafiltration of blood. Second one is a modification of this extracellular fluid by the peritoneum, and last one is cellular contribution, which will be carried out later on. If we talk about ultrafiltration from blood, coelomic fluid is derived from blood plasma as well as interstitial fluid. The mechanism is fairly simple. There is ultrafiltration occurring from the capillary walls and coelomic epithelium, and modification is happening by the action of peritoneum, which we say mesodermal lining or mesodermal in origin. There is secretion of specific substances, some ions and some proteins, and even specific cells. After all the addition of these substances, we call this fluid as coelomic fluid, because this is a now modified and well regulated filtrate, and this filtrate came from blood plasma as well as interstitial fluid. Now let's talk about the composition of this coelomic fluid. We have three kinds of composition, which is basic composition. Then we will talk about the cellular components, what kinds of cells are there, and in last we will talk about miscellaneous components, which are taxon specific and vary greatly. Let's talk about the basic or fundamental composition of coelomic fluid. There is 90% of water, and water acts as a medium of transport, and it will also act as medium for hydrostatic support. So it will be forming the hydrostatic skeleton. There is introduction of electrolytes, and electrolytes include sodium, potassium, calcium and chloride ions. And what is the function of these electrolytes? They will maintain the osmotic balance, they will perform osmoregulation, and they will also be generating the membrane potential. If we talk about proteins, proteins are somewhat in lesser concentration than the blood plasma itself, and proteins are of various types. There may be some kind of transportation proteins, some immune proteins, some kind of enzymes. There may be presence of nutrients, and nutrients include basic glucose, amino acids and lipids, and all these nutrients has to be metabolic in nature. And if there are nutrients, there is metabolism, there has to be metabolic waste, and the metabolic waste is of what kind? That will depend on the type of organism and its habitat. It may be ammonia, it may be urea, it may be uric acid, carbon dioxide is always there. If we talk about the cellular components, the cells are called coelomocytes, and there may be of different types like amoebocytes, phagocytes or some kind of granular cells. They will function accordingly. There are certain cells which are functioning for the wound healing and wound repair. There are specific cells for the transport of various kind of nutrients, various kind of materials, metabolic wastes as well. And if we talk about taxon specific additional components, there may be various kinds of pigments. Example, we can take hemoglobin like substances in some annelids. There may be some kind of signaling molecule for coordination between different organ systems. There may be some specific enzymes as well. What is the role of this coelomic fluid? The role of the coelomic fluid is determined by these components itself, like hydrostatic function, because there is presence of water and electrolytes. There will be osmoregulation as well. So fluid is incompressible. It will provide a rigidity which will enable the movement, and it is very much crucial for soft-bodied animals like annelids. It acts as a transportation medium for nutrients, for gases like carbon dioxide, for metabolic waste products. And if the circulatory system is primitive type or not evolved at all, then it becomes very much important, and it becomes very much the sole medium of transportation for all these essential components. There is a function of lubrication which will reduce the friction among various organ systems, between organ and organ, between organ and gut wall, between organ and body wall, and it will enable the smooth internal movements of that organism as well. It will maintain the internal body space and distributes the mechanical forces acting on that organism, because organism has to move into various places within its habitat. There is immunological role which is present in many coelomates, and the main role is phagocytosis of various kind of toxins or foreign objects. There is the role in defense overall. There is the role in storage of these nutrients. Sometimes storage of gametes will also take place, storage of waste product before they are released or diffused out. Now come to the classification of eucoelomates, and we will classify eucoelomates on the basis of mode of coelom formation. So there are two types of organisms based on this mode of coelom formation. either these are schizocoelomates or these are enterocoelomates. What happens is sometimes the mesoderm will split into certain pouches and those pouches will be forming the coelom. In such a case, the process is known as schizocele, and the organism in which this splitting of mesoderm is forming the coelom, such organisms are called schizocoelomates. Whereas in some organisms, the coelom is formed by the outpocketing which is occurring in the cells of archenteron, and archenteron itself is a part of the endoderm, but the cells which are outpocketing will be converted to mesoderm so that the coelom will be formed. And in such an organisms where the coelom is formed by outpocketing of archenteron, we call such organisms enterocoelomates, and process itself enterocele. As you can see in this diagram, there is splitting within the mesoderm, and this splitting is creating certain pouches, and these pouches will be forming the future coelom, and such a process is schizocele, and the organism is schizocoelomate. Whereas in this second figure, you can see this is the endoderm layer and this is the archenteron itself, and there is outpocketing in the archenteron. Endodermal cells are performing these outpocketing. Do not get confused by the endoderm, because what happens that these outpocketing cells will be differentiated into mesoderm first, and after that there will be formation of coelom. So coelom is still outlined by mesoderm. That is why it is a type of eucoelomate, which is true coelom lined by mesoderm from all the sides. So let's talk about schizocoelomates. The term schizo means split. So there is splitting of mesoderm which is occurring to form this coelom. Mesoderm is forming a solid band, and this solid band will split internally to form the coelom, and the cavity will appear within the mesoderm itself. So this is a typical feature of protostomes. Protostomes are the organisms in which blastopore will develop into mouth first, and the typical examples include Annelida, Arthropoda as well as mollusca. As you can see, there are some solid patches which will be splitting, will be forming the coelom itself within the mesoderm. Now let's talk about the process of schizocele itself. So schizocele is the formation of true coelom by splitting of a solid mesodermal masses into two layers, creating a fluid filled cavity. That is very much obvious. As you can see in this diagram, there is the formation of the solid masses within the mesoderm, and there is splitting occurring inside the mesoderm internally, and this splitting is leading to the formation of these pouches, and these pouches are but will be forming the coelom in the future. If we talk about the steps directly, there is the stage gastrula, where the formation of three germ layers, ectoderm, endoderm as well as mesoderm will take place. Mesodermal bands will be forming which are the solid masses, as you can see here, and there will be internal splitting within these masses, and this internal splitting will create a cavity within the mesoderm. As you can see, these are the small cavities which are forming by the splitting of mesoderm. This splitting will result into the formation of two specific layers. The one is somatic mesoderm, which is also called parietal mesoderm, and second one is splanchnic mesoderm, which is also called visceral mesoderm. The somatic mesoderm will line the body wall. It is toward the outer side. As you can see, outer side is the somatic mesoderm, whereas inner side is the splanchnic mesoderm, because the splanchnic mesoderm is surrounding the gut itself, and together these two layers will be forming the peritoneum, which is also known by the name coelomic epithelium. After that, there is the formation of mesenteries. It takes place in the regions where layers remain connected, and at last there will be maturation of this coelom. By maturation, we mean that the cavity will be enlarged. There will be the formation of coelomic fluid through the process of ultrafiltration. There will be maturation of this coelomic fluid by inserting down various electrolytes, by inserting in various coelomocytes, various cells that will be performing the various functions. There is maturation of coelom, and finally we get the proper coelomic cavity, and at last the organs will start developing and maturing in this space. So this is the process of schizocele. In diagram, as we already described, there is the formation of these presumptive mesodermal masses. Splitting will take place internally. The inner side will form splanchnic mesoderm, outer side will be forming somatic mesoderm, and these cavities will start expanding. The ultrafiltration will allow the fluid to rush in. There will be maturation of this cavity. There will be insertion of various kinds of electrolytes, various kinds of nutrients, various kinds of coelomic cells, and finally we will be getting this cavity we call as a coelom. Mesenteries have already been formed, and organs now will be start developing inside this cavity further. Now let's talk about enterocoelomates. By entero, we mean gut. The coelom will come from the archenteron itself, from the gut itself. Coelom will be formed as the pouches from the archenteron. So archenteron will outbulge and there will be formation of some pouches. So initially these cells are from the endoderm, but these cells will be differentiated into mesoderm so that it will be forming a true coelom which is lined by mesoderm from all the sides. So these pouches will pinch off and they will be forming the coelomic cavity in future. And this type of coelom is typically seen in the deuterostomes. Deuterostomes are the organisms in which the blastopore will give rise to anus first. Let's talk about the process of enterocoeli. As you can see, first of all, there will be the stage of gastrula where the formation of all the 3 germ layers have been completed, and now there will start outpocketing of the archenteron. As you can see, the outpocketing of archenteron has been started. So the lateral wall of archenteron will form certain pouches that will start to outbulge into the blastocele. So here this cavity is the blastocele and these some cells from the endoderm are starting to outbulge. Now there is separation of these pouches. Soon these pouches will start to look like a separate kind of pouches. We can say there is pinching off from this archenteron and now they will exist as independent sacs, and these independent pouches will be forming the future coelom. Now there will be mesodermal differentiation. So these cells of these pouches will be converting or will be finally maturing into mesoderm for the purpose of formation of peritoneum, which is coelomic epithelium, which will be required later on. Once the mesodermal differentiation or mesodermal maturation has taken place, there is the formation of 2 layers. Now this is very similar to schizocellus development. The 2 layers, the somatic mesoderm as well as splanchnic mesoderm. The somatic mesoderm is what lines the body wall. The splanchnic mesoderm is what lines the gut. After this, mesentery formation will take place. At final, the maturation of this coelomic cavity will occur. The cavities will enlarge. Multiple cavities may fuse to form single cavity in many cases, and there will be filling of this cavity with coelomic fluid. There will be addition of coelomocytes into this coelomic fluid. Organs will start to develop inside this cavity. These are the outpockets. Outpockets have started to pinch off. Now these endodermal cells will mature into mesodermal cells, and after that multiple cavities may fuse together. There is formation of the splanchnic layer from inside, and outer layer will be forming the somatic mesoderm. Finally there will be maturing of these small pockets or one large cavity, we can say, where the fluid will rush in. There will be development of proper organs. What is the major difference between the schizocoeli and enterocoeli? If we talk about basic mechanism, in case of schizocoeli, there is splitting of solid mesoderm. There are patches of solid mesoderm have been formed, and splitting between those patches, the internal splitting is taking place to form the schizocoelomate organism. Whereas in case of enterocoeli, there is outpocketing within the archenteron, the outpushing of archenteron, which will lead to the formation of enterocoelous body plan. If we talk about the origin of coelom, the cavity is appearing within the mesoderm. Whereas in case of enterocoeli, the cavities are arising as archenteronic pouches. They will differentiate into mesoderm and finally mature coelom. If we talk about process in short, the solid mass is splitting and forming cavity, whereas the pouch is forming, the pouch is detaching, and after that the cavity is forming and maturing. Where is this schizocoeli found? It is found in the protostomes, whereas enterocoeli is found in the deuterostomes. Examples include Annelida, mollusca, arthropoda, and in case of enterocoeli, the most common examples are echinodermates, hemichordates, and all the chordates. Let's talk about coelom in case of arthropods. If we talk about arthropods, arthropods are eucoelomates, means there is true coelom which is lined by mesoderm from all the sides, and there is fluid filled inside this true coelom, that is coelomic fluid. The difference between arthropods and other true coelomates is that in arthropods the coelom is highly reduced. The coelom is reduced to the regions of gonads and some excretory organs, structures like nephridia. So we can say that in case of arthropods, coelom is not the main body cavity. The main body cavity in case of arthropods is what we call haemocoel. What is this haemocoel? Haemocoel is the dominant body cavity, or haemocoel is the large body cavity which is filled by the hemolymph. Haemocoel is derived from the persistent blastocele cavity, and it combines with the extended spaces of open circulatory system. Now let's talk about the haemocoel formation in case of arthropods. Just like normal development in case of arthropods, the gastrulation will take place, 3 germ layers will be made, the mesoderm will form, the solid masses of mesoderm that will be formed, and internal splitting will take place. The process is called schizocoely. There will be formation of true coelom. This true coelom will be reduced only to the regions of some excretory organs and gonads. In the rest of the body, it becomes reduced and very discontinuous. There will be development of open type of circulatory system. Blood vessels, if present, are directly opened into the sinuses. These sinuses will expand greatly. They occupy most of the body. What is meant by open circulatory system? The vasculature is very much reduced and organs are in directly contact with the circulatory fluid. So what happens is the blood vessels will open into sinuses and will bathe the cells, tissues and organs directly, and these sinuses will start expanding greatly and occupy most of the space of the body cavity. We can say that Haemocoel is an indirect extension of open circulatory system in case of arthropods. Along with this process, there is persistence of the blastocelic cavity, and this blastocelic cavity will also start expanding. So both open circulatory system spaces or sinuses as well as this expansion of blastocelic cavity will contribute to Haemocoel formation in case of arthropods. Why is the coelom reduced in case of arthropods? Because of development of this open circulatory system. So an open circulatory system, there is open hemolymph circulation which led to expansion of Haemocoel. This expansion of Haemocoel also contributed to reduction of the true coelom. What are the other factors which are contributing to reduction of coelom in case of arthropods? The other factors include very rigid exoskeleton, some joint appendages, there is efficient tracheal system in insects as well, there is a lesser need for hydrostatic skeleton, the largest coelomic space is also not required. The exoskeleton is fulfilling the need of this hydrostatic skeleton. The well-developed tracheal system is performing the function of respiration very efficiently. So what is the role of Haemocoel? The main role of Haemocoel is that it has replaced the prominent roles of coelomic cavity. It will act as the dominant cavity in case of arthropods. So it will be performing the function in the circulation of hemolymph, because it is the extension of the circulatory system itself, and there will be transport of nutrients, transport of hormones, transport of metabolic base. All those functions will be performed by the Haemocoel in place of true coelom. And what is the role of reduced coelom? The maturation of gonads will take place. Functional physiological role of this reduced coelom in case of reproductive physiology of these arthropods will be there, and there will also be excretory compartmentalization. There will be some excretory function as well. At last, let's compare Eucoelom with Haemocoel. If we look at definition, Eucoelom is a body cavity filled by the coelomic fluid and surrounded by mesoderm from all the sides, whereas Haemocoel is not surrounded by mesoderm from all the sides, but it is a blood-filled cavity, or we can say it is filled by hemolymph. So it is a blood-filled body cavity which is forming the main cavity in case of arthropod-like organisms. Origin of Eucoelom is obviously mesoderm. It can arise through the process of schizocele or enterocele, whereas in case of Haemocoel cavity, origin is dependent on the persistence of blastocele cavity as well as expansion of the open circulatory system or expansion of sinuses of the open circulatory system. Now let's talk about the mesodermal lining. So Eucoelom is completely lined by mesoderm, whereas in case of Haemocoel, there is no such lining by mesodermal epithelium tissue. Peritoneum and mesentery are the exclusive features of Eucoelom. Both these features are absent in case of Haemocoel cavity. So what is the nature of fluid? Nature of fluid in Eucoelom is obviously coelomic fluid, which is plasma-like and very much regulated. Fluid of Haemocoel cavity is hemolymph or other kind of circulatory fluid if the organism is different. Now what are the functions of fluid in case of these Eucoelom and Haemocoelom? The functions are basically hydrostatic support, transportation, the same function in both the type of organisms. If we talk about the arrangement of organs, so in case of Eucoelom, the organs are well suspended and compartmentalized, and in case of Haemocoel, compartmentalization is not that much and organs are freely bathed in the hemolymph or circulatory fluid itself. If we talk about compartmentalization and hydrostatic skeleton, they are well developed in case of Eucoelom, whereas these are poorly developed in case of Haemocoel type of organisms. If we talk about circulatory system, so Eucoelom will be perfectly supporting the closed circulatory system. Either it is fully closed or it is partially closed, whereas Haemocoel is exclusive for open circulatory system. If we talk about control over internal environment is highly controlled, highly regulated because of the presence of this peritoneum, whereas in case of Haemocoel, there is very low control control. In case of Eucoelom, the primary body cavity will be formed as Eucoelom, whereas in case of Haemocoel, at embryonic stage, the Eucoelom is formed by the Schizocoelom process, and later it is reduced to those excretory as well as reproductive areas. There is expansion of circulatory sinuses. There is persistence of the blastocoel cavity, and both of these will contribute in the formation of Haemocoel cavity. Arthropods Eucoelom was formed during the embryonic stages, and it will be greatly reduced to small remnants in the reproductive area as well as the excretory area. Whereas in case of Haemocoels, this will become the dominant body cavity and persists in most of the body part. That was all about the concept of coelom. That's all for this video.